in addition to being quite a cutting edge surgeon, literally, and no pun intended, you're also known to be really an outstanding researcher in the field and have some really novel knowledge on some of the most current techniques in understanding cells and doing that research. So you're the expert. So I love to hear directly from you and not from me trying to put it all together.
Yeah.
So could you give us a little update on the ongoing synovial sarcoma biomarker study that's going on at Levine Cancer Institute and what you hope to discern from that project?
Of course. So, and to review the concept of looking for biomarkers or trying to find some pieces of information about somebody's tumor that can tell us about how their ultimate prognosis, how they may respond to treatment, are they at increased risk for the tumor metastasizing or increased risk of dying from it. And so we've had a previous successful biomarker studies here performed, such as the liposarcoma study. So we next decided to turn our attention to synovial sarcoma, and this project has been building more or less since I came here. Synovial sarcoma is an interesting type of sarcoma because the genetic features, or I should say the molecular features that it has, the changes that are made to the cell's DNA in order to make it a cancer, is a pretty unique mechanism compared to a lot of other sarcomas. The changes that occur in synovial sarcoma essentially they have to do with the packaging of the DNA. So it's not that the DNA is having individual mutations to genes that are either making them more active or turning them off. And it's not that the DNA is having big chromosomal changes, which is that's the way that a lot of sarcomas change their genomic information in order to become a cancer. Synovial, pretty much all has to do with DNA packaging. And because of that, we think that there are susceptibilities to treatment that this may not work for all sarcoma patients, but it's a really intriguing idea to look into. And unfortunately, when you look at the literature, there haven't been that many large-scale studies evaluating the molecular characteristics of synovial sarcoma. The largest study was published in 2017 from the Cancer Genome Atlas, the TCGA adult soft tissue sarcoma study, where they looked at approximately 270 patients, and then they did comprehensive sequencing. So that, by that I mean they look at what is happening on the DNA level. Are there mutations? Are there changes in the structure of the chromosomes? They look at the RNA, so not only what DNA exists in the cell, but what of that actually gets turned into the message to make a protein. They look at other structures such as microRNA, as well as the DNA packaging. So that study, well, it was very interesting, only included 10 synovial patients. So we, especially with the wonderful records keeping and specimen preservation that Dr. Ahrens has helped us out with, that have approximately 75 specimens here at this institution. Some of them are from the same patient, but nonetheless, that is a wonderful amount of specimens. And so we're in the process, we're near completion of the protocol in order to get this started, but we're essentially performing the largest, most comprehensive multi-platform sequencing of synovial sarcoma yet performed. And then we intend to use that information. So we have patients that had evidence of metastasis and patients that did not have metastasis. And so we're going to compare these genetic changes and try to identify, can we deduce what is going on in the genome that puts some patients more at risk than others? And then we eventually have the goal of using that information to help guide us and decide, are there special medications that we can give patients to help treat them? So this is kind of the starting block for a number of different projects that can come in the future.
That's really exciting, and to think it's such a large study. And as we both know, in sarcoma, it's very hard to gather a large group of biopsies or a large group of patients for clinical trial enrollment because of the rarity of these subtypes. And so you don't hear the word large very often with sarcoma,
Absolutely.
That's really exciting.